Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2024. This research sheds light on a newly discovered genetic cause of a severe muscle disease in a cat, offering valuable insights into the diagnosis and understanding of this condition. The study focuses on X-linked dystrophin-deficient muscular dystrophy, a type of muscular dystrophy that is inherited through the X chromosome and results in the progressive weakening and degeneration of skeletal muscles, ultimately leading to a fatal outcome. This disease is caused by variations in the DMD gene, which plays a crucial role in maintaining muscle integrity. In both human and veterinary medicine, accurately diagnosing muscular dystrophy is paramount. In human medicine, early genetic diagnosis allows for the timely initiation of management strategies aimed at slowing the decline of muscle function. Current guidelines recommend genetic testing for boys showing typical signs of dystrophinopathy and elevated levels of a muscle enzyme called creatine kinase (CK) to confirm the diagnosis. While genetic testing for muscular dystrophy has not yet been widely adopted in veterinary practice, the researchers emphasize its importance in accurately identifying the disease. Establishing reliable genetic tests could also help prevent a serious complication. Some cats with dystrophin-deficient muscular dystrophy can develop a life-threatening condition called rhabdomyolysis, which involves the rapid breakdown of muscle tissue, after undergoing muscle biopsies under general anesthesia. Therefore, the development of less invasive diagnostic methods is highly desirable. Currently, in feline medicine, the primary genetic testing methods available are traditional techniques like polymerase chain reaction and fluorescence-based Sanger sequencing. These methods are effective for detecting genetic variants that are already known. However, when it comes to identifying new or novel genetic changes, more advanced techniques like next-generation sequencing, NGS, are more suitable. Next-generation sequencing allows for the examination of the entire genome, or large portions of it, making it possible to discover previously unknown genetic variants associated with diseases. Several studies utilizing next-generation sequencing have successfully identified both known and novel genetic variants linked to various genetic diseases in cats, including other forms of muscular dystrophy. To date, 5 different variations in the DMD gene have been identified as causes of X-linked dystrophin-deficient muscular dystrophy in cats. These include 2 large deletions and 2 so-called nonsense variants, all associated with a severe form of the disease resembling Duchenne muscular dystrophy in humans, as well as 1 missense variant linked to a milder form similar to Becker muscular dystrophy. Given that there are already 5 known target variants to consider when testing for X-linked muscular dystrophy in cats, and the DMD gene itself is very large, the researchers highlight that next-generation sequencing is a valuable tool for both detecting known variants and uncovering new ones. In this study, the researchers aim to identify a disease-causing genetic variant in the DMD gene by employing whole-genome sequencing, WGS, on an affected cat. Furthermore, they evaluated how common this newly found variant was by analyzing the genetic material of the affected cat's parents and a large group of cats from a breeding colony. The research team reported on a 10-month-old male Kinkolo cat, a breed resulting from a mix of American Curl and Munchkin breeds, that was diagnosed with dystrophin-deficient muscular dystrophy at the Hokkaido University Veterinary Teaching Hospital. This cat was initially brought to the hospital due to persistent increases in serum liver enzyme activities. During the diagnostic workup for suspected chronic liver disease, which included a physical examination, blood tests, X-rays of the chest and abdomen, and an abdominal ultrasound, the cat surprisingly showed no obvious clinical signs of muscle weakness or gait abnormalities, and its tongue appeared normal. However, blood tests revealed markedly elevated levels of alanine aminotransferase, aspartate aminotransferase, and significantly, creatine kinase, an indicator of muscle damage. Thoracic radiographs and abdominal ultrasound and computed tomography scans revealed abnormalities in the diaphragm, the main muscle involved in breathing, suggesting a muscle-related issue. Based on these imaging findings and the elevated creatine kinase levels, a muscle biopsy was performed to obtain a definitive diagnosis. Tragically, just 2 hours after recovering from the anesthesia for the biopsy, the cat suffered a cardiopulmonary arrest, suspected to be due to severe rhabdomyolysis, and passed away. Post-mortem examination of the muscle tissue from the abdomen and diaphragm showed significant abnormalities, including variation in muscle fiber size, degeneration, necrosis, calcium deposits, fibrosis, and the infiltration of immune cells. Importantly, while the liver tissue appeared normal, immunohistochemical staining revealed a complete absence of dystrophin protein in the affected cat's muscles, confirming the diagnosis of dystrophin-deficient muscular dystrophy. In contrast, dystrophin staining was clearly detectable in a control muscle sample from another cat. To identify the genetic cause of this cat's condition, the researchers performed whole-genome sequencing on the affected cat and compared its genetic information with publicly available data from other cats. They also used two different versions of the domestic cat genome as reference points. One derived from an Abyssinian cat, known as FelCat9, and a newer assembly from an American short-hair cat, called NAMS 1.0, to account for potential differences in genome assembly. Their whole-genome sequencing analysis of the affected cat, along with comparative analysis against 43 other cats, identified two genetic variants that were predicted to have a high impact on protein function using a software tool called SNPFF. One of these variants was a stop-gain single nucleotide nonsense variant that was detected in both the FelCat9 and NAMS 1.0 genome assemblies. This type of variant introduces a premature stop signal in the genetic code, leading to a truncated and likely non-functional protein. Specifically, this nonsense variant was located within the DMD gene and affected multiple transcripts, which are different versions of the genetic information that can be produced from a single gene. The researchers noted that in both genome assemblies, over one-third of the DMD gene transcripts were predicted to be shortened due to this variant. The second high-impact variant identified by SNPFF using the FelCat9 assembly was a one-base pair deletion. However, this deletion was found to be present in all 44 cats included in the whole-genome sequencing analysis when aligned to the FelCat9 assembly, suggesting it might be a common variation or a potential error in this particular genome assembly. Interestingly, this deletion was not detected when the same sequencing data were aligned to the NAMS 1.0 assembly. Further investigation using Sanger sequencing on the affected cat, its parents, and several cats from the Abyssinian and American Shorthair breeds confirmed the absence of this deletion in all tested individuals. These findings strongly indicated that the one-base pair deletion identified in the initial whole-genome sequencing analysis using the FelCat9 assembly was likely a false positive result due to a misassembly in that particular version of the cat genome. The researchers pointed out that the region where this deletion was seemingly present in FelCat9 contained a stretch of repeated guanine bases, which are known to be challenging to sequence accurately, and can lead to errors in genome assembly. The fact that the newer NAMS 1.0 assembly and Sanger sequencing did not detect this deletion suggests that the NAMS 1.0 assembly provided a more accurate representation of the DMD gene sequence in this region. Crucially, the researchers performed Sanger sequencing to validate the identified nonsense variant in the affected cat and to determine its presence in the cat's parents and a large breeding colony of 354 cats, including 97 kinkolos, 125 American curls, and 132 munchkins. The results of this targeted sequencing confirmed the presence of the nonsense variant in the affected cat. However, this variant was not found in either of the healthy parents, or in any of the 354 cats from the breeding colony. This absence in the parents, who showed no clinical signs of muscular dystrophy and had normal physical and neurological examinations, blood work, and imaging results, strongly suggests that the nonsense variant in the affected cat arose spontaneously. Such spontaneously occurring genetic changes are known as de novo variants. The fact that this variant was only present in the affected cat and not in the extensive breeding colony further supports the conclusion that it is a rare, newly arisen mutation likely responsible for the cat's muscular dystrophy. The researchers also noted an interesting pattern. The newly identified nonsense variant, like other previously reported single nucleotide variants associated with X-linked dystrophin-deficient muscular dystrophy in cats, was a C to T, or G to A on the opposite strand transition. This type of genetic change is known to be the most frequent form of de novo mutation in the cat genome, suggesting that the DMD gene might be particularly susceptible to this type of change. The discovery of this de novo nonsense variant adds to the growing list of genetic variations in the DMD gene known to cause X-linked dystrophin-deficient muscular dystrophy in cats. The fact that the parents did not carry the variant highlights the importance of comprehensive genetic testing, especially when dealing with suspected inherited diseases where parents show no signs of being carriers. The study underscores the utility of whole-genome sequencing as a powerful tool for identifying novel genetic causes of disease, particularly in cases where traditional genetic screening methods targeting only known variants would fail to detect the underlying mutation. While traditional genetic tests in cats often focus on known single genetic variants using methods like Sanger sequencing, next-generation sequencing approaches can screen much larger portions of the genome, making them ideal for identifying previously unknown variants like the one described in this study. Although next-generation sequencing can be more expensive than traditional methods, the costs are decreasing, and more targeted next-generation sequencing approaches focusing on specific genes like DMD may become more widely accessible in veterinary medicine in the future. The researchers believe that the integration of genomic data with electronic health records and large-scale databases will be crucial for advancing precision medicine in domestic cats, allowing for more accurate diagnoses and potentially personalized treatment strategies. The study also highlights the importance of utilizing high-quality and well-annotated genome assemblies for genetic research. The discrepancy observed with the one base pair deletion between the two different cat genome assemblies used in this study demonstrates that genome assembly quality can impact the accuracy of variant detection. The researchers concluded that in the case of the DMD gene, the ANAMS1.0 genome assembly appeared to be more accurate than the FelCat9 assembly, possibly due to the presence of repetitive sequences in the FelCat9 assembly that can lead to errors during the assembly process. This finding emphasizes the need for ongoing improvements and comparisons of different genome assemblies to ensure the reliability of genomic analyses. The researchers acknowledge that their study focused primarily on the DMD gene and that future investigations are needed to conduct more comprehensive comparisons across other regions of the genome between different cat genome assemblies. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.